Morning. I don't think we can hear what you're saying.
Сергей Васильевич, здравствуйте. Мы вас не слышим. Джейкоб, я напишу кураторам. То, мне кажется, Сергей Васильевич нас не слышит тоже.
Хорошо. Хорошо.
Да, сейчас, секунду.
So let's go back to the new algorithms we have for the molecular docking techniques, and one of these algorithms which claim to be very powerful and very efficient in terms of the clustering is the diffdock, which is based on the diffusion model, reverse diffusion model, and was created in the MIT and is using some kind of diffusion machine learning algorithm. And here we are trying to compare different machine learning models with the standard molecular docking techniques. Also, there are some regression models for docking. And the problem is that the uncertainty, epistemic uncertainty is kind of philosophical term. It's like kind of epistemic uncertainty because when you implement the randomization in the movement of the ligands, you will create some kind of uncertainty of the ligand movement, which is producing a lot of distortion and steric clashes. Here also, they try to solve this uncertainty, but still produce the distortions and the steric clashes in a way to generate the right pose. So the pose is not really taken into consideration. This is actually the drawback of the diffdock. You will have the receptor, and it's not really processed as a crystal as the atomic structure. It's processed as a kind of a diffusion model. So you got the receptor and you got the protein ligand binding site, and this protein ligand binding site it was processed to produce some kind of a diffusion map. So I will talk about it later. But here we would like to point out that there are some algorithms like equibind and a tank bind. You probably try to use it as well, together with GNINA machine learning algorithms. So far, we get four main machine learning algorithms, which is GNINA, diffdock, equibind, and tank bind. And a diffdock is the best in terms of the clustering. Empirically, diffdock obtains 38% top 1 success rate with their MSD less than 2% angstrom on a PDB bind database, significantly outperforming the previous state-of-the-art of traditional docking and deep learning methods. 23% and 20% respectively. So it's a good result. In terms of the clustering, as I already mentioned, the clustering means when you have the pose, the experimental pose, the experimental reference as a ligand in a protein ligand binding site, you would have a matching as a superimposition of the model against the reference. And usually the deviation from the initial structure is, initial means the reference from the initial structure is less than 2%, 2 angstrom. So, which means the confirmation of the model is very close to the confirmation of the co-crystallized ligand. There's some kind of a stereochemical validation. So you, whatever molecular docking you would like to do, it's always good. I mean, this is, I would point that out that this is necessary to do the stereochemical validation in terms of the validity of the molecular docking. Otherwise, if you didn't really validate the method, you don't know whether it's really performing well. And this validation was in many studies was neglected. So people are just skipping this part, not validating, not producing the right RMSD, and they were trying to convince everybody that it's working. It's not really working because no validation. So we don't know. Sometimes it's hard to do the validation because you don't really have a co-crystallized ligand. Then you would try to produce something like homology model. You will try to do some kind of the ligand alignment to see whether the binding sites are kind of artificial. You will do some tricks to create the model which is close to the crystal model. Then you will do the molecular docking. Then you can do some sort of experimental validation. It's not really experimental validation, but at least your data is derived from the experiment, so you can claim that you are actually successful with the stereochemicals, with the validation, with this post-validation technique. And right now we have this diffdoc, which is very good at this validation step. So whatever protein you have with the co-crystallized post, you will do the diffdoc with the trained with the pre-trained system and do the molecular docking, and it's good, and it works very well. At least I tried to already do some kind of benchmarking with the previous structures which were published by me last time. So most of the time the search-based molecular docking techniques fail to give me the right success rate, but using this diffdoc algorithm, I was successful to produce the very good alignment. And diffdoc maintains significantly higher precision, almost 22%. Therefore, we can say that diffdoc has a fast inference time and provides confidence estimates with a high selective accuracy. So what are the details of this diffdoc algorithm? Well, the idea is to use the reverse diffusion to produce some kind of translation, rotations, and torsions of the ligand, and to see how you could actually rank poses and using the confidence score. As I already mentioned, docking produced significant epistemic uncertainty, and a lot of methods will inhibit these uncertainties about the correct pose between multiple alternatives. Regression models to minimize the square error, they are good, but they're not very efficient, as we see here. So we have the protein. This is the diffusion model of the protein, the representation of the protein using the diffusion model. and you can see we get some kind of a representation of the binding site. So here we have the program also treats some part of the protein as a binding site as well. So you get one binding site and the two binding sites where the ligand sits, with the two bound structure sits. So this is a kind of a reference structure, and this is the binding site generated by the regression model. And then we do some trick to perform the molecular docking, just removing the noise of this diffusion model, trying to match our pose with the reference ligand. Actually, it's some kind of artificial. So here it's not really the optimization algorithm. When you have the randomized moves, you are trying to achieve the right position in where the binding site is located. So those of you who is already doing some experiments using molecular docking knows that in a standard protocol, you have the binding site, you will generate some kind of a grid where your ligand is located, where your co-crystallized ligand is located, and you will try to do the docking, randomizing the moves of the ligand of your ligand of interest. And after some iteration, the energy will decrease, and hopefully you will have the ideal match, at least close to the reference, the match of your model to the reference model, to the experimental model. Here we have some kind of a pre-trained model of the binding site here, which is located here, and by reducing the noise, we are matching artificially. So we're kind of reducing the searching space, the search space of the system, and by doing this, we are matching the generative samples to our two bounds structure. So the next benefit of the regression model, that they are also minimizing the square error mean of this technique, which is also good. And generative models will populate most models and will then predict the right structure among these. So this is the benefit of the generative models also used in a TIFF-doc. So this is the information on the product space diffusion. As I already mentioned, this is a kind of space. As you can see, it's a kind of here we are talking about the 2D space. We have to do it in a 3D space, and by calculating tangent space and improving the alignment of our models. So the diffusion generative modeling works on the manifolds, providing the score model predicting the target space. A lot of mathematics here, so I'm not going into which vectors are rotating, or usually important to retain the model. So it's not really what we have in a conventional search-based methods, because in the search-based methods we have the atoms, we have the bonds, we have the torsional angles, and then we just specify these angles, torsional angles, to make them move in a way that's specified by the genetic algorithm, for example. But the problem is that in the past, people were trying to implement deep learning methods. They failed to outperform search-based techniques. They used the equibind and the tangbind as a regression-based deep learning models. Actually, they didn't really succeed on the significant speed up over the search-based techniques, because the search-based techniques are relatively fast in comparison to the deep learning models. Okay, so let's just have a break, and we can start in probably 10 minutes, okay? To use the new technique with the improved efficacy, and for that reason, this was the DiffDoc. In the DiffDoc, they implemented the confidence model to rank and evaluate sample confidence. And what is it all about? What is the confidence, and why we are going to use it? Because in a standard traditional molecular docking methods, we are usually using the parameterized physics-based scoring function and search algorithms. The scoring function usually takes the 3D structure and returns the estimate of the quality or likelihood of the given pose, while the search stochastically modifies the ligand. So it will modify the positions, orientations, and torsion angles, with the goal to find the scoring function of the global minimum. So this is the idea of the standard molecular docking. On the other hand, these search-based methods remain computationally not efficient and inaccurate due to the limitations concerning the searching space. Usually the searching space is very limited. The standard searching space is the kind of cubic searching space, up to 60 angstrom for the autodoc. For the autodoc winner, it's 2 times less. It's the searching space, the cubic searching space for the autodoc winner is about 30, 25, 22 angstrom. And therefore, this might create some significantly impaired results when present with apostructure. So you have the apostructure and the holostructure and the apostructure when there is no ligand in the holostructure. When you have the ligand, and you will try to perform the experiments with the apostructures when there is no ligand present. But of course, if you optimize if you perform the validation of the method, you know where the binding site is. You will put the grid into the center of the protein ligand binding site, and we'll try to perform the molecular docking. And sometimes there is a kind of ab initio search when you don't really have the information on the binding site. Then you can specify the whole protein and ligand just popping through this protein in different crevices and pockets, and to have a chance to find the some kind of a global minimum of this interaction. For this reason, this is very hard to perform the blind molecular docking, because searching space is very limited, and we need something more efficient. On the other hand, the machine learning techniques for blind docking have to tackle the blind docking task by directly predicting the pocket key points on both ligand and the protein and aligning them. So it's a kind of a trick they use. First they predict pocket key points, and then trying to use it, train the system and to use it to improve the efficacy. For instance, the tank bind technique improves over this by independent predicting a docking pose for each possible pocket and then ranking them. However, this one-shot and few-shot regression-based predictions are orders of magnitude faster. Their performance has not yet reached the traditional search-based techniques. As I already mentioned previously, despite this, the benefits, we didn't really improve the efficacy, the performance of the molecular docking. On the other hand, the diffusion generative model, using some kind of a confidence score, so it's not a physics-based score, it's a kind of a score which will give you the information on the alignment, so whether the alignment is good, or if the alignment is not good, the score is also, the confidence is not good, so probably the efficacy is not good. And this approach learns the distribution over the Euclidean space with the 3D coordinates, rather making them not very good suited for molecular docking if you will use the standard scoring function. So then, if I will sum up this information, so I would just give you some highlights here. That the confidence predictions are very useful for downstream tasks. We can train the confidence model to take the generative model samples and return the score for each sample. Samples are ranked by the score, and the of the best is used over overall confidence score. So as you can see here, we have different, the score can be also positive and can be also negative. Like this, the score is positive and the score is negative, positive too, and which means that it is in this binding pocket that probably have the most of the true binding structures sitting here, and you get the other binding site when you would have here, which will give you the negative score. So what is the scoring model? The scoring model is described here. There are some vectors. There's two principal vectors where the ligand is actually flexible. you're performing the translations, rotations, and torsions using these two vectors, and to update the ligand binding poses. So they're using some kind of tangent space with the C3 symmetry, programming these positions, orientations, and torsions as shown here, and you get some kind of pre-trained model. Then what they did, they just took the database, pre-trained it, had the information concerning these parameters, and then used this for the ligand of interest. So then you can just apply it for your own system. So, or you can if you have some kind of a new system, like it's not a protein ligand but something else, then you can just take some database, train the model, and try to implement this information on your system. For instance, if it's some kind of ligand cyclodextrin interaction, probably it will not work on the ligand cyclodextrin interaction. So those of you who is interested in a Nature or Science publication, this would be also very good to investigate how DiffDock performs on the cyclodextrin molecules. There are also some crystal structures, and one could also implement this information in a protein, in a cyclodextrin ligand to obtain the high accuracy. Although the cyclodextrin is very small, and if you will provide enough sampling to the system, you probably would achieve also good results using the search-based techniques. On the other hand, if we have some sophisticated systems like some polymers, functionalized polymers with a very complex topology of the surface, and then we would like to have a interaction like we already did with Anastasia, using the hydroxyapatite surface and the interaction of some ligand on the surface. Then, of course, this might be the benefit of the technique. I would also try to do the diffraction analysis to come up with some idea how the poses actually interacting with the surface to produce at least ONMR to produce the experimental validation for the DiffDock. But anyway, it's also good to try this new technique, and the ability of this new technique, at least on these materials, which are not really included in a classical simulation like protein. So this would be also very good approach if we will try to implement it for beyond the protein ligand interaction and trying to use it on the material science structures. So, and what is the workflow? Workflow is you have the embedded with the RD kit for to manipulate the ligand. The ligand could be also provided with as smiles. Then you convert it to the 3D representations. Then you sample the random poses, and then you simulate reverse diffusion. So you kind of you have a protein, so you sample some number of random poses, then you will populate this number over this protein, just make it populated with the ligand in a different directions. And then you simulate the reverse diffusion. As I already mentioned, the reverse diffusion takes into consideration the information about the protein ligand binding site, and gradually you reducing the noise, considering only the poses which are located in the protein binding site relation. So finally, if you will populate, so program just rejects all the unnecessary ligands, because it will only focus on the area where the reference binding site is located, and will provide only the poses which are very closely matched to the reference ligand. And then you got you have to rank and select top poses. Top poses to specify, at least to have some idea on the information of the efficacy in terms of the binding, which is very closely observed, which is very closely in terms of the 3D alignment to the reference ligand. Okay. As I already mentioned, this kind of good technique in terms of the clustering, but again, you don't really have a good scoring function to identify the efficacy of the binding efficacy of the ligand of interest. So the standard, what are the standard benchmarks which are used for this new algorithm? They did the PDB binding. They used the standard benchmark PDB binding database, and this database contains about 20,000 experimentally determined structures of small molecule plus proteins. So they just took them and they trained the model and compared the results with different algorithms. They also took the different algorithms and did the same on that. And as you can see, there are some very old, some old algorithms like SMINA, or its derivative of VINA. So it was published 10 years ago, and some are very relatively new, like NINA and TankBind, Equibind and TakeBind, just published a couple of years ago. So quite efficient. Here's the algorithm which was developed by Schrödinger company. It's a commercially available algorithm. And what do we have here? We have the machine learning algorithms. They're more efficient, but again, they produce the results, which is only 30% of predictions with the RMSD less than 2 angstrom, which is not bad. It's like a third of the molecules is matching to the experimental structure. On the other hand, if you will use diffdoc, you will increase it to 10%, and it will be close to 40%, which is quite good in terms of the performance. So you could say that diffdoc outperforms the search-based deep learning algorithms, especially the old algorithms like SMINA, for instance, or even the GLIDE is not really very productive at this point. That two times higher productivity was achieved by using this new algorithm. Okay, so this is what is all about the DiffDoc, and I could also mention that if you would like to use the DiffDoc, you have to install it on the cluster, and you have to use the CUDA accelerator, the GPU accelerator to run this algorithm. On the other hand, there are some enhancements in this algorithm, so people are also adding some kind of a scoring function to this algorithm using Nina, which is the fork of Smina, but with improved convoluted neural network scoring function, which is physics-based scoring function. So you can actually see that the ligand, which is highly possible, will bind to the receptor according to the orthodox, has also the high binding energy in terms of the Wiener results. So if you will combine these results in one, then you could have at least some kind of a scoring function. But again, you have to run two software sometimes. You will not see the good correlation and the regression plot. And this would also produce some problems. On the other hand, DiffDoc is not really very selective to the protein ligand interaction in terms of the steric clashes. You get the steric clashes, and then these steric clashes would create some disturbances in the post-processing of the pose. So you don't know what kind of pose you would take. That usually you would take the Nina pose and not the orthodox pose. Then it's kind of a discrepancy. So there are a lot of problems so far that should be addressed to improve the pipeline using the orthodox, using the DiffDocs. So sometimes people are creating some kind of scoring functions like a hybrid scoring functions or empirical scoring functions. So they're processing the libraries, like binding databases, and trying to produce some kind of calculated IC50 values, IC50 base scoring function, or constant, the binding constant, the dissociation constant dependent scoring function, which is, again, creates a lot of concerns how these scoring functions are feasible, because there is no validation. I was actually supposed to improve some algorithms. So people were trying to do such techniques, but they didn't really use any validations, just created something and claimed this is working. Usually it's the case for small startups and pharmaceutical companies. So they can create in some kind of pipelines. 99% of these pipelines don't work because there is no validation. They don't do the benchmarking, the right benchmarking. So this is just junk, the piece of junk, and that's why so far, if people are combining machine learning models with the standard search-based model, sometimes it also creates a lot of discrepancies. So we don't know what is exact scoring function we have to implement. On one hand, we have this very sophisticated confidence. On the other, we have the scoring function. Sometimes they not really correlate very well. So it creates a lot of problems. But again, diffdoc is good in terms of the prediction of the RMST less than 2%, but how it will really promote the information about the accuracy, the binding is a big question. So we have to do a lot of efforts to improve this drawback. So as I already mentioned, they used NINA computational algorithm, and in NINA you have the receptor and the ligand, and you specify the binding site where the receptor and the ligand would meet, and you would provide the exhaustiveness, the standard exhaustiveness function when you apply it already in a Wiener algorithm using Monte Carlo chains. And after that, you will have a full refinement. Then you will do the rescoring. Actually, you already know what the Monte Carlo chain is. So I explained you last time the Monte Carlo algorithm, and then they perform the full refinement and do the rescoring and sorting the pores using the convoluted neural network scoring rescore function, which is available in Python. So they just improved the rescoring technique as a final step of molecular document. Created some kind of a hybrid technique. This is a standard searching based technique, but on the top of that you got the rescoring function, which was machine learning, which was enhanced by the machine learning algorithm, something like that. Yeah, but I didn't really test it. Here we see that NINA and SMINA are very close, actually very close. Maybe you have some improvement, like 5%, 4% improvement, but the improvement is not very significant. So, but there is some improvement. Well, one could see that at least 5%. You will have a 5% improvement. I don't know whether it's really feasible for different types of the proteins, like dimers, or it's just a monomer ligand interaction. They don't really specify what kind of protein complexes they use for their purposes. Okay. So, and I also would like to give you some introduction on the projects we have concerning the molecular docking techniques, and some projects were done in the past by me. This project is not really finished. This was unfinished project, and those of you who is interested in a simulation of protein ligand complexes, we got this nice system which represents the protein ligand structure. Here you have the protein which is called the HIV protease. The protease means that this is the enzyme expressed by the virus, and this enzyme is important to produce the mature protein for the viral life cycle. The viral life cycle is a very complex process, and you get some kind of polyproteins, because the genetic material of the virus is extremely small. So the viruses, they are trying to save the space, and they don't really have enough space for the open reading frames, for the standard open reading frames, as we see in the eukaryotic cells. And you could have a transcription in the different directions. Therefore, it would create different proteins like a polyprotein. So one protein is translated as not as a one protein, but also the two proteins, they are translated as one, or three proteins, they are translated as a one big protein. To say this is just an evolutionary example how the viruses would try to save the space of the genetic space in terms of the size of the capsid. Therefore, when they translate this polyprotein, this protein has to be chopped up into different fragments, and these fragments are matured protein. And this process is done by using the HIV protease. Therefore, if we will inhibit this HIV proteases, the virus would become impaired and ineffective. So we would create some cure to treat some viral diseases like the HIV. So this was already known for many years. If we were trying to inhibit the HIV protease, we will have a potential to treat the HIV patients. And as such, we have this very nice HIV protease structure. It is created by two monomers, so it's a dimer. It will be only efficient as a dimer, and the dimer has a catalytic site, as shown here, and this catalytic site is a kind of a protein ligand binding site, but it's not really a protein ligand binding site, but we call it a catalytic site, created by the aspartic residues. So if we try to inhibit these aspartic residues in a catalytic site, then we would impair this protein structure, and the virus is not really active anymore because there is no packaging, or the polyprotein is not processed, and the virus cannot be cannot produce the viral, cannot be packed into the virus-like particles, or into the virus particles. So this is the idea, and the viral particle is impaired. And what we have here for this project, we have the HIV protease, and we have the inhibitor, which is called indinavir. And indinavir is the already well-known inhibitor, was discovered in the last century, and it was found to be effective against the protease, but with time there is also tolerance and the resistance to this drug, and the virus could escape the detrimental effect by using this substance. So this project was published, partially was published by me when I had the complete indinavir in the HIV protease complex, and we used hollow structure, ligand bound state as a receptor. You can obtain it from the PDB database as an X-ray model. It's also very nice to model because you have a symmetry. It's C2 symmetry, very symmetrical. So you could actually see very nicely the catalytic site and whether the structure is open. Sometimes you have these flaps, and when these flaps are elevated, the structure is kind of open. It also can be closed, so it's very good model to simulate using different techniques like molecular docking or molecular dynamics, when you can have this movements of the flaps. And you can also have information on the binding site where the ligand is sitting. So here you can see there is different representation. This is a cartoon representation. You have also molecular surface, and the grid can be also added to the protein ligand binding site. Therefore, this model also can be used to be analyzed using the DIFTOC algorithm or similar models. I also modified this model when we do the iteration of the one part, the translation of the one part of the protein, when the one monomer is rigid and the other monomer is translated, then you can you can actually specify the binding. You can analyze the binding as a kind of a release, the ligand release, some sort of a transition state release of a ligand. So the ligand would migrate from the standard binding site to the binding site created by one of the monomers. So by using, of course, you will have some kind of S-like curve which corresponds to the pharmacodynamics, and the S from this S1, from this S curve, you can calculate the transition Gibbs free energy, which is very, very precise, because in the molecular docking you have an uncertainty, and from this uncertainty, this would create the differences in the binding energy up to 1.5 kilocalories per mole, which is very, very high when you convert it to the inhibition of the constant, inhibition constant, up to nanometers, micrometers range. So sometimes it's extremely hard to calculate this parameter because the standard error is very high. So by using this technique, you would come up to the method to improve, significantly improve this parameter. Okay, let's just have a break, and we will start in 15-20 minutes. Okay, let's go back to the projects we have, and projects where we are using the molecular docking technique. So I was talking about the protease, the HIV protease as an example. this protease has two monomers to create one dimer. This is a homodimer. It has a catalytic center, and it has aspartic acid residues as an active in its active site. And it also has a C2 symmetry, which makes it very good candidate for molecular modeling, analysis, polar apostructure, conversion, and molecular docking validation and improvement. So I would definitely use it as a good candidate to validate different molecular docking techniques. So this is the example to give you the idea what are the standard molecular docking methods like search-based methods are capable of in terms of the evaluation of the protein ligand efficiency and bioactivity of various ligands. As an example, Indinavir anti-HIV substance to inhibit HIV protease. This is the protocol, how we use in this protocol to analyze and to molecular docking using the autodoc. So here I have some kind of a pipeline to analyze, and I will give you the introduction. When you have the autodoc installed, you can just open the PyMol and download the HIV protease here. You could use different representation of the protease like this. This is the stick model, line model. You can add also the cartoon model, specifying what type of cartoon you want to implement. Like this is a tube cartoon. You could also use stick models. As you can see, this is the Indinavir structure as a stick model, and you will try to perform the molecular docking, specifying the grid parameter. These are the grid points, such as present here, and this is the center of a grid point. You have to make sure that the grid has a right dimension. The dimensions are usually 60 angstroms, but you can modify it according to your binding site, as present here. And you would assign the grid space. The grid space is 0.3 angstrom to do the molecular docking. Then you have to generate the protein as a compatible structure, as the autodoc compatible structure, the ligand, and then you would run the autogrid first to calculate the grid maps, the atomic grid maps according to the force fields used for different atoms in the autodoc. So as you can see, the grid maps are calculated. Then you will start to do the autodoc, and the autodoc is usually takes about a couple of minutes, and the standard protocol generates up to 10. This is the up to 10 various up to 10 runs. So usually you are specifying the number of runs, and these are the 10 runs, and from these 10 runs you could extract the information about the ligand. So as you can see, we have the pose, which is not really very well suited to the ligand, to the reference structure. If we will rotate it, the information about the pose is the best. So it's -10 in terms of the binding, which is quite good, but the alignment is not very good. And therefore, and this is just information about the grid map. So you have different grid maps for the carbon, for the oxygen, for the nitrogen, for sulfur, if you have a sulfur atoms in a molecule. And usually it's very well suited for the small organic molecules without any heavy atoms in their structure. And here is a good example how you can actually perform the molecular docking. You can actually specify different ligands and run it in a script using some standard script protocols. Then you can analyze the scripts. But this pipeline is also good in terms of the validation. You can just do the validation and observe in the PyMol how your structure is validated according to the reference. So as you can see here, the reference is not really very well matched to the best pose, and therefore the optimization of the protein and the ligand is important. Here we didn't do the optimization, we just used X-ray crystals. Sometimes it's hard really to improve the quality of the crystal because there are different resolution models of the X-ray crystal structure. Usually the best resolution is about from 1 to 2.5 angstrom. Sometimes you have bad resolution structures, and sometimes even you have some NMR studies when you have complexes which are in the solution, and you get different ligand conformers, so you have to choose the right conformer in order to perform the docking. It's also not quite easy to do, and for this protein we didn't really have multiple conformers of the reference drug, but we had the crystal structure, which is probably not very well optimized for the protein ligand interaction. Therefore, there are some standard protocol for the physical chemical validation, like stoichiometric validation. We have to remove the steric clashes. Usually the protein structure is not very clean, and one should produce the clean structure using different algorithms. You can use Schrödinger maestro. You can use Moyer, which is also very powerful. You can use some open source protocols like VADA protocol, more probity protocol. Sometimes it's important to check the stereochemistry of the protein. You can use ProCheck, and from this analysis you can have more information whether you need to improve the quality of the structure. Usually when the quality of the structure is good, you are able to improve the alignment of your ligand on the receptor, on your alignment of your in silico pose on the experimentally derived pose. So this is the information, some molecular docking result from my previous publication more than 10 years, 15 years ago when I published it when I was a student, PhD student in Germany. So this is the kind of optimization. This was the optimization of the indinavir ligand. We were trying to implement the pharmacophore similarity scoring function using some standard algorithms. At the time, I was using the Sybil algorithm. Sybil algorithm is very well-known algorithm, and even now you have very powerful chemoinformatics techniques like COMSIA analysis, but it's not really available anymore, and we have it, and it's also possible to do some 3D QSAR studies. So at that point, we generated a novel hit, which is shown here. But when you perform the molecular docking, sometimes it's not really very well suited in terms of the improved energy of this novel hit. So first, you have to derive the binding site, the protein ligand binding. Devise the protein ligand binding site, then you will do the for this particular experiment, I did Molegra virtual screening. So this was done using Molegra molecular docking technique. So when I performed this in Molegra, I actually came up with a higher affinity for the novel hit. As you can see, there are some aspartic residues which are actually inhibited by using the propofol, by using the indinavir molecule, and the same holds true for the novel hit as well. So these aspartic residues here, you can see they're also inhibited, which means that we actually succeeded with the possibility to inhibit the aspartic sites. And so if you will have a look at the novel hit here, so you got 3 aspartic sites which are inhibited according to the color corresponds to the different monomers. So this is the monomer 1, this is from one monomer, and this is from the monomer 2. So all of these aspartic residues, especially aspartate in the position 25 is inhibited. And here, when we would use indinavir, only 2 aspartic residues are inhibited. So which means like this, aspartic acid residues, when we are inhibited, 25, 29, and 30. So as you can see, we get 25 and 29. But here we also have additional aspartic site, which is also inhibited. And we can say that even from the analysis of the protein ligand binding site, we could say that this molecule probably is more efficient in terms of the amount of the aspartic site inhibited by this molecule. Also, we have the additional molecule, additional residues, isolysine here, glycine here, and there is a lysine here, and which is also inhibited, and we can also analyze the distances, the hydrogen bonds between the oxygen and the hydrogen atoms in the protein ligand binding site. So one could also notice that the hydrogen bonds occurred at the distance from 1.7 to 2.2, roughly it's 1.7, 2.2, and here this ligand is producing the hydrogen bond at very short distance. The distance is 1.7, where here we have the hydrogen bonds at the distance of 1.9 to 2.1 approximately. So, however, we need additional analysis, and I'm not really presenting this analysis here, but you could also notice that different algorithms are giving you the different number of depends on the processing of the binding site. You sometimes you have to specify the distance from the ligand at which you would analyze the hydrogen bonds between the ligand and the receptor. And here also some aspartic residues when we analyze the indinavir, here we observe that the number of the residues is lower than in comparison to the novel heat. This is also kind of a information you have to understand the binding affinity, the inhibition affinity of the ligand in comparison of the novel heat in comparison to the reference ligand. And here's the pharmacophore model we were trying to analyze. As you can see, also the pharmacophore model kind of really closely related to the indinavir structure. So if you would just analyze the pharmacophore, you can see the centers, the charge centers, and positively charged the hydrogen bond donors and hydrogen bond acceptors, together with the lipophilic centers, are quite similar here. And this can be also explained by the similar activity of this novel heat. So one could also do the geometric analysis on the pharmacophore, but we didn't really do this analysis, and I would also give you some information on the best pose when I was trying to perform here using the standard Autodoc algorithm. As you can see, when you have the best pose and the worst pose interaction, then there is no matching here, and so actually I didn't present it here, but this is really hard to do the matching using the Autodoc experiment. And when you compare the novel heat, you will have slightly lower energy in comparison to the indinavir, which was what the case using the molecular algorithm, which was already. That means that the optimization is not really good. And when you can compare the experimental data you would observe that the experimental data of Indinavi is very diverse. You will have differences in orders of magnitude, like observed here. You have 0.05 to almost 400 nanomolars. It's 0.4 micromolars, which is very huge. And when you can convert it to the inhibition constant, you will have at least for this value, you would have at least 67. Of course, this is the representation of the binding value, which is here. So it's also in the range of this experimental inhibition constant. But again, this is really hard to compare because the energy is quite huge. The inhibition constant is quite huge, but when you compare to the novel heat, you will have 67, which is 2 times lower than the reference. So I would like to point out the difficulties to analyze the protein-ligand interaction using the search-based standard binding molecular docking models. So at this point, one could devise something like presented here. You got it's NINA. It's a NINA pipeline. When you got standard technique, you specify the box here and in the receptor, and you can add ligand and do the Monte Carlo chain simulation using the particular exhaustiveness value, applying the Metropolis criterion. The Metropolis, I already mentioned the Metropolis criterion in the last lecture, and how you can reject or accept some ligand moves according to this Metropolis criterion. Then you will have some kind of full refinement and the rescoring and the sorting, the post-rescoring and the sorting. And then finally, you will have the RMST filtering to produce the final result using improved CNN scoring functions and enhanced CNA scoring refinements to increase the accuracy in 5%, which is also the improvement. But as I already mentioned, we don't really notice the significant improvement of this technique. However, if we will perform something like diff-doc scoring improvements, then we could probably, because diff-doc is already known to produce the very high matching between the reference and in silica and the results, then we could just improve the scoring function, and then we will produce and we will increase the accuracy up to 40-50%. So I would like to mention I would like to say a couple of words concerning the molecular docking in the synthetic biology and how we can implement it. But just a few sentences, a few words, because I will give a presentation I will give a lecture in the next week or so, what we have in terms of the synthetic biology, how we can actually implement molecular docking techniques, why it is important, what is the synthetic what the synthetic biology is used for, the computational synthetic biology, or the experimental synthetic biology. Right now, we have also the PCR machine. So those of you who wants to mutate different genetic materials also is welcome to embark on different scientific projects, so we can generate some RNA, DNA molecules to screen, to create some very sophisticated scavenging systems, nanorobots. I'm not sure what is the limitation of this technique, but it's actually endless. So whether you are interested in this kind of projects, I advise you to meet me, and we can discuss what can be done in this direction. And here's just one example. We are using the protein-protein interaction, molecular docking protein-protein interaction analysis to understand how we can actually treat the cancer cells using information on some mechanisms known as chromatopsis. So I would discuss it in the next lectures in more detail, but as you can see here, we also have proteins. Some proteins are important to induce the apoptotic event in a cancer cell. So we are using them to match the experimental data. So this is the experimentally derived data. And we are trying to model what exactly happening with some proteins in the cells, how they can be mutated, and how this mutation impairs the stability and the function of different protein when the chromatopsis is taking place. Okay, for today, thank you for today. Thank you for your attention. This is all for today, and I'll meet you in the next lecture. If you have any questions, I'll be happy to answer.
Yes, can you hear me?
Yes.
All right. First, thanks for the very interesting lecture. And I have a question about actually the practice related question. When you showed the launching Autodoc procedure, there were like kind of 10 runs, and the 10 corresponding scoring function values. And the question is actually, does that mean that you pick the 10 runs, or the algorithm decides to stop on 10 runs? And this decision is derived from the scoring function value. Well, basically the question itself is like, when the molecular docking algorithm decides when to stop. So
yes, you can specify the number of the iterations you have to produce. You can also specify 100 iterations for the for the genetic algorithm, for the molecular docking technique. Depends on the protocol. For systems like cyclodextrin ligand, it's a good idea to improve the genetic algorithm iterations, like runs, up to 100 probably, because the system is quite small, so it's better when you increase in the similarity search. Probably you would come up with a more accurate result. But for the proteins, the standard procedure is to run the protein the algorithm for 10 runs, for 10 iterations. Yeah, and therefore you will just specify it. There are also numbers of mutations, crossovers provided in a config file. There is a config file. You will put all the information to this config file, and the system is running according to your specifications. You just have to program these numbers, but usually people are not really modifying these values, because this is the standard algorithm. You just run it and see what is the result. But of course, sometimes you could improve the runs. You can increase the number of these runs by increasing it. Of course, you have to run then for the significant amount of time, because for 10 runs it would take approximately 5 minutes. Yeah, maybe up to 5 minutes. So if you will increase the number, you would just increase it, and you will just have to run for 1 hour just for one protein. This is sometimes not really feasible. If you have let's say 100 ligands, so you would probably run for many days, for 4 days, and this is not very practical. Therefore, if you will have some kind of a standard protocol, then you will know that this is just for 3, 4, 5 minutes, then it's okay. Yeah, because of course, I mean, you can only also run for 1,000, but then this will increase the computational cost. So this is the idea, to optimize this value, to make it not very, very long, and at the same time it would be accurate enough to have the meaningful result. In my case, I'm usually using 10 algorithm runs, genetic algorithm run. It's called GA run function, genetic algorithm run, and GA-run. And for the host-guest interaction, I would increase it to 10 times. So.
Oh, and sorry, the question gotta be stupid. I've never done it before. So one run is basically like one position, like one position with somehow mutually interacted, like ligand and protein.
Yeah, one run is
or it's like a search of all positions.
I don't understand what I mean by the position. You have the protein and you have ligand, and the protein and the ligand starts running, starts moving, and starts to increase the kind of trying to set up its volume, molecular volume in the binding site. So it tries to adapt itself in a protein ligand binding site, because the space is limited. Yeah, so we have some kind of conformational matching. We will provide the ligand, it rotates within the binding site. You will specify different iterations in one run, so it's like you repeating it. You're stopping this run, then it stops after some iterations. This is a couple of 100,000, I think, of iterations. So it's just running and stops. Then you will have the second run and the third run and so on. Yeah. So it's like it just rotates and produces the number of different iterations. So it means the run when the algorithm starts working. In terms of the specifications, you will have the number elitism, you will have the number of mutations, you will have the crossover range and things like that. Yeah. Then when you specify all of this, the ligand starts moving.
When it moves near the binding site or all over the surface of the protein?
No, only in the binding site. You're going to specify the binding side, the grid box, in the grid box, actually, the grid box is the binding side. Then it starts moving, yeah? So it actually
yeah, no, I got
moves,
it. Uh-huh.
yeah, it actually moves 10 times, and then you will process this information. So you will have some kind of a pause, free, it will produce when it moves, it produces the energy. So you will have some kind of a total energy, like the minimal energy. From which run you have the minimal energy, for instance. Like I will show you here.
Oh yeah, so out out of 10, like repeated
Yeah,
iteration,
here,
you
like
pick
this.
the minimum one.
Yes, you have here, you have 10 runs. Yeah, for instance, you have 10 runs, and from each run you will have the minimal energy. Like minimal energy for 10, for one run you have this energy, then you have this energy, and then something like that. And for the first, it's actually ranking. It's kind of
So it's not chronological order, it's just like a rank.
no, it's not. It's
Yeah,
not really. It's not really the first run, the second run. It's just, the run is going like randomly. Yeah, it could be like seventh run, fifth run. It's just, and after that you will do the ranking, because the run is producing some kind of energy, some kind of a minimal energy, like this, yeah, or like this. And then once you get 10, some kind of a 10 results, then you will do, the algorithm is doing the ranking. And you got the rank, the run is different. Here would be the fifth run or something. Then the rank is,
got it.
yeah, something like that, yeah. I also have the script where you can also put all the information, like the rank and the run number, and this is the free energy, then the pause number. This is also the pause number, just the same as the ranking. This is the artificial, of course, the number. Yeah, and that's it. So you will just combine, and if you will add 100 runs, you will have 100 values here.
Oh yeah, yeah.
Here, that's it. Very simple. Yeah.
Oh
The
yeah,
runs
yeah,
that
no, I got it.
corresponding, the runs that corresponding actually the ranking and the number of runs are the same. You will just increase the conformational search, the conformational search of the system. More number of runs, more exhaustive is the algorithm, let's say. That's it. So you
Oh
will
yeah,
have,
thank you very much for the answer. Yeah, no, now we completely got it. All right, and then so provided that we have like 10 here, we have 10 relatively short runs. So would that mean that we have like big number of sort of cheap calculations rather than a small number of expensive ones? Why I'm asking whether we should kind of launch it on the CPU or GPU.
no, this is it runs on a CPU, this algorithm, this particular algorithm, because it's not the diffdog. The diffdog works differently. This is a genetic algorithm, and genetic algorithm is mostly done on a CPU. If you use Autodoc Vina, then it's parallelized, so it's quite fast. So it would increase the speed in the order of 10 times, more than 10, I think 10 times. So if for the standard algorithm like this, you need at least 5 minutes, or from, let's say, from 1 minute to 5 minutes, depends on how big your ligand and how big is your receptor. So usually for the ligand it's important, because the ligand then rotates, and sometimes it will increase the amount of time drastically. Yeah. So for this, you just need one CPU. Then you would run it for 1 minute. If you will use Vina, it will give you the information in a seconds, maybe, I don't know, 10 seconds, from 5 to 10 seconds per ligand. So you can apply it in a high throughput, in a virtual screening. Yeah, but this one is not really good for, you can actually apply it for the virtual screening, for the molecular docking up to, let's say, 50, up to 100 ligands. So if it's more than 100, it's really hard to do the molecular docking using the AutoDock. Or you have to compile it using some, I think there is some GPU already, GPU version of the AutoDock. Probably it's possible. But again, this is not really very easy. That's it, but it works when you have the small number of ligands, and it's fine. Then you can just produce the numbers and compare to your system. Okay.
Okay, and one small question. So the calculation you've been showing during the lecture, you performed it on your own computer. So these are not like really expensive.
No, these are no.
Not in some cluster or something.
No, this is my result. We have the diffdoc, which is compiled on the GPU, but this is also possible to run using SSH. So, those of you who is interested in the AutoDock, with the diffdoc, you can compile, you can run it on a GPU. But this one is possible to run on the laptop.
Oh yeah, perfect. Okay, yeah, just I was curious whether I would be able to test it like on my own computer. And then
Yes.
Okay,
yes.
yeah, great.
You could use the same protocol. I think Anastasia will help you how to install it, and it's relatively easy using the Linux system. So
Okay.
that's it. I think if you have some other questions, you can just ask me.
Okay, last small question, and yeah, I promise we're done. I've been searching for the software, like something really like for the newbies, like to test, and I found the Verachem VDoc software. Have you heard of it? And can you say anything about it? Or we should start with AutoDock right away.
I also have the software, but the problem, the Verachem is a Petachem. So this is the kind of molecular dynamics software when you will run it on a GPU, but it has the kind of outdated GPU system, and if you're talking about Petachem, but if you're talking about Verachem is the software for computer-aided drug design, right? So it's just a commercial company. I also have the software. I downloaded it when it was free of charge, and they just they have redoc, they have redraw, reconf, they produce the confirmation, we charge, we filter. Actually, it's okay. I mean, you can use it. They also have some kind of semi-empirical quantum mechanical potentials, but they also integrated it into the MOE graphical user interface as a VM2 platform.
Yeah,
I think if they have the academic license, I'm checking now. The package, it's Verachem. It's called Verachem. They have a commercial license, 1 and 2 year commercial license, and they also have the academic license. Then you can use it.
Yeah, that's what I'm asking. They have like,
but I'm not sure whether they can provided for you as you are in Russia?
Oh, well, actually, it's they have like everything for academic users, except for VDoc, which you have to specifically request from them with the explanation what what would you like to use it for.
So you already installed this, yeah?
So like my nationality, my citizenship didn't interfere anyhow. But so I'm asking about whether we should use VDoc. So I would text them and ask for this, or we'd better start with Autodoc right away.
No, the Autodoc is a kind of a classical thing. You can also,
Okay.
there are like 50 different molecular docking protocols. The VDoc is also probably, I didn't test it, but you can try to test it and compare, for instance. Yeah, you will have the Autodoc, which is free of charge. And right now it works very good in our project. But you can also use this VDoc. Sometimes it's also good to use different algorithms, like 3-4 algorithms to check the validity of your experiment. That's it. So I don't really see any problem. I mean, if you really want to use, I actually have like probably full, like more than 20 different protocols, different software modeling pipelines, including also, I could also use VDoc. I just didn't use it, but probably it's fast protein ligand docking and scoring, and maybe it's also good. But as far as I know, I mean, right now, DiffDoc is better in a way that it will increase the conformational sampling up to 20%.
Okay. Yeah. Thank you. Thank you very much for the detailed reply. Yeah. No, I'm done with my questions for today.
Okay.